Thank you. And I'm not sure who wrote the title for that, but I'm going to touch on several of the things we talked about at the conference, because Neuralstem is sort of a walking example of having to deal with all of them. Just real quickly, we are a public company in the U.S., and I'm going to be doing all this in the context of giving you an update on some of our clinical trials, so I will be making forward-looking statements. Again, for those of you not familiar with Neuralstem, our technology are human neural stem cells. These are regionally specific. This is a fetal-derived tissue, but it has a mitotic capacity of about 60 doublings. So, for instance, if you go down to the spinal cord cells here in the middle, those are the cells we have in humans right now. The single GMP-approved cell bank in the U.S. will be able to transplant every patient in the world that we ever need spinal cord cells for. So this is what we do. We actually have 2 regenerative medicine platforms. We transplant the cells into patients. We also have, and I do want to spend a little time today talking about it, a novel small molecule drug, which is, we believe, the first truly neurogenic drug. It's neurogenic specifically in the human hippocampus. We know this because we can grow hippocampal neural stem cells, human in dishes. And this is in now a second trial to treat major depressive disorder in the U.S., going into all these other indications also. And I think a harbinger of the future of regenerative medicine. But I want to start with ALS, which they call motor neuron disease over here. Last week in Europe, our principal investigator, Eva Feldman, talked about the trial data from the first trial, 18 surgeries. These are the world's first intraspinal injections. So the cells go directly into the gray matter of the spinal cord, not into the intrathecal cavity or the fluid. And we thought that the results are very encouraging. First, just to give you a feel for what we do, what we're doing, and this is, yes, the human spinal cord here. I don't know if this is working or not, but we'll see if it does. Right. So what you're seeing here is what we call a floating cannula. The hard part goes in and then pulls back because the spinal cord actually has a little bit of a vibration or a pulse of its own during surgery, and you don't want a hard needle inside which can shear the cord. And what we're doing is this is a targeted surgery at each segment, and in this patient, this was an early patient that was only transplanted here, we're actually putting the cells into the motor neuron pools at these segments. So this is where the cholinergic motor neurons die in ALS patients. And so the processes that go out and innervate the muscles die and the muscles atrophy. And that's the disease. And when the muscles that control breathing and swallowing die, the patients die. On average, about 3 to 5 years from diagnosis. There's nothing that either increases significantly their life or improves their quality of life. And so we're putting in our spinal cord cells. We have excellent data that showed that the cells in animals actually synaptically integrate. So this is not a transient. We expect these cells to graft and live for the entire life of the patient. We did have 6 patients, very late-stage patients, who died and volunteered for autopsy during the trial, and there is very good data, DNA fingerprinting, that the cells, in fact, do survive the entire lifespan of the patient. So this is what we do, and when people talk about transplantation, as you can see, we have to do a little laminectomy here. You open up the cord. This is a very invasive. This is not minimally invasive. But I will tell you that when we budgeted this trial originally, to get back to the practical aspects of doing this, we figured the patients would be in a ICU for a week and then a step-down unit for a week. All of the patients went home after 3 or 4 days, none with any weaknesses or problems that were a result of the surgery. So we're very pleased with that. Now, this is the device which was invented by our surgeon, Nick Boulis, who at the time was at the Cleveland Clinic. You can see the cannula and the syringe going in. This is actually attached to the patient, not to the bed frame, so that if someone moves by accident, again, you're not going to shear the cord. We probably did 100 pigs to prove to the FDA that this was safe. So again, the practical points here, when you do these surgeries, you have to do them in a place where if something goes wrong, the patients can be treated, right? There's a lot going on here. You can't just do this anywhere, even though there's nothing really exotic for neurosurgery in the injections. Again, this is just to give you a little bit of the scale. This was our star patient who went public and had a remarkable recovery, which is something you rarely see. But I want to talk to you, again, Eva Feldman, who is the overall PI. What I really want to talk about is the way we did the trial, and some of the data. What we're doing, remember, is not replacing the motor neurons that died. We're putting in cells in the motor neuron pools to support and nurture back to health the remaining cells. And so the FDA required the first 6 patients to be very late stage. 2 of them were already on breathing machines. And sort of by definition, also they required us to only do lumbar surgeries for the first 6. So the fact that they were non-ambulatory and late stage sort of by definition meant that we couldn't help them. There were not enough cells to rescue there, especially with the minimal dose they let us do in the first trial, which was 100,000 cells per injection and 5 or 10 injections, 2 different groups of 3. Once we showed that was safe, they let us move up to ambulatory patients who by definition should have enough remaining motor neurons to be able to nurture, maybe bring some back to health. And so what you're looking at here is patients 7, 8, and 9 who received 5 lumbar injections, but they were ambulatory. And what you're looking at here is, this is obviously a non-responsive patient. Now, there is no control group in this trial, and I didn't put up sort of historical controls, but this is kind of what they look like for ALS patients. And so what you're looking at here, this is the ALS FSRS score, which is the clinically validated efficacy score, actually. So we're at over 800 days here, and you can see these patients are remarkably stable. This is something that's extremely encouraging and has never been shown before for ALS patients. And then this is patient 10, 11, and 12. And again, this is Ted, since he's gone public, we can say. And he's actually back up here from down here to normal. But again, you can see clearly, now these guys were done later. We were only allowed to do 1 patient a month, so the timelines are not quite as long. Here's the surgery. But again, with these 2 slides, what we're showing for the first time ever is really slowing the progression of the disease and or actually improving the patient over a very long period of time. Now, these patients, in addition to getting 10 lumbar injections instead of 5, also came back and received 5 cervical injections. So they had 15 injections for a total of 1.5 million cells. There are lots of individual components to the ALS SFRS score that measure breathing and all kinds of things. We are going to focus in the next trial on cervical injections. I think you see some data here. Basically, the FDA is going to allow us to go up to 400,000 cells per injection and 40 injections per patient at the end. So the cervical patients will go up to 20 injections, 10 on each side, and from 200 to 400,000 cells. And then the last 3 patients will again come back, and they'll get 20 lumbar injections. So we'll be going up to 16 million cells from 1.5 million. And we think, actually, that this is a more is better story. Now, there is a real estate problem here, right? Because you can put too many cells in and impinge on the spinal cord. Remember, these patients, even though they may not be able to walk, are not paralyzed the way spinal cord injury patients are. There is signal coming through, and they can certainly feel pain. So you have to be very careful, and all of the animal studies were targeted at that to make sure. We believe that this will be the maximum safe tolerated dose, and that will be ultimately the therapeutic dose that we'll be delivering, which will be 400,000 cells per injection, 40 injections, 20 cervical, 20 lumbar. Ultimately, of course, we're going to have to go into the brain also to really treat these patients. But for what we're seeing here, holds up in larger studies, and we expect that it will. It looks like the take-home message from this trial right now is that we can help early-stage patients. Eva also presented data showing that the bulbar patients, which is where the onset comes first from the top down as opposed to usually from the bottom up, which is a small percent, didn't do well in the trial. So the patients that did the best were the ambulatory, earlier stage patients, non-bulbar. And that's a tremendous result, considering the dosage that the FDA limited us to. So we have just recently also gotten FDA approval to do spinal cord injury with the same cells, the same device. In the U.S., the FDA has approved us for chronic spinal cord, which is defined as basically a year to 2 years out from the injury. It's interesting, you heard some earlier panels talk about regulatory attitudes. Even though we've been approved now to do 40 injections with 400,000 cells per injection in ALS patients, and ALS patients are all breathing compromised. They're in many ways much more fragile than the spinal cord injury patients. The FDA gave us the exact same protocol where we will start with lumbar patients in the spinal cord injury, do 8 patients, these will be complete patients, Asia A, and then wait 6 months, and then assuming they see it safe, they'll let us do the cervical spinal cord injury patients. At that time, we will already be doing the 20 injection, 400,000 cell per injection treatment of the ALS patients, probably at the same hospital. But this is kind of their mindset in terms of the escalated risk, the way they do these trials. We are also going to do acute spinal cord injury in Seoul at 4 different centers. We have a partner there, CJ Sheljadang. And again, you heard panels earlier talking about harmonization and, regulatory issues. Neuralstem, we're also going to be doing a stroke trial in China starting in the next few weeks. Same cells, direct injection into the brain. So we're doing cell therapy trials in China, Korea, the United States, and probably this year also in the Mideast and in Mexico. And to be honest with you, we are the harmonization. We are imposing our own FDA-approved protocols on all of these markets. We are imposing our own, FDA-approved, the CMC component manufacturing everywhere we go. And I think that is the reality of how this will work. Certainly different countries, different cultures look very differently. So we only deal with incurable diseases. And the FDA, as you know, takes a very conservative approach to how you can do that. They're trying to change this. You heard Michael Werner talk about the new regulations and how they've been sort of encouraged to try and look at these diseases differently. I will tell you that a month or so ago, they held the first of their public hearings on this for ALS. And many of our patients and supporters showed up, and it made a difference. Within 2 weeks, we had our FDA approval for phase 2. We had a very aggressive approval, as I said, up to 400,000 cells per injection. Now, again, as you saw, the phase 1 data, which they had seen, is as good as anybody could have ever hoped for, and very clear, right? I mean, it's not hard to look at that and say, okay, 700 days out, these patients are clearly improved or stable, and even these patients, 800 to 900 days out, right? They don't look like this. So from our point of view, obviously, we had hoped to get that, but I think that the take-home message there is that as you move through various indications, the patient advocacy groups do hold a lot of power. And they can bring a lot of pressure to regulatory groups and probably to payors eventually. I think we're going to end up with that same dynamic as we move into commercialization. We are, as I said, doing the stroke, direct injections into the brain. This is basically 4 months out, so this is chronic motor disorder from stroke. And again, moving to China, there was a time, so we built our own manufacturing facility there, again, using the CGMP protocols from the FDA. We have not been anywhere in the world, and this literally goes from China to, Europe to Mexico, where the regulations are tougher than they are from the FDA. So there are always things that you have to address and get through. But basically, if you come in with the real high quality, it will be accepted everywhere. The interesting thing about China, of course, gets into the question that you all were talking about yesterday, which is reimbursement and payors and pricing. So we have the same product now in human trials. And in China, by the way, this is a combined phase 1, 2, 3. And at the end of 120 patients, if the data permits, we'll actually be commercializing in China. And so we're talking about, regulatory issues, payors, reimbursement issues in multiple indications now across multiple continents and different cultures. And there are lots of issues involved, but they're just that, they're issues. And you deal with each culture in each country as you go in, and you can't let it stop you. I don't think there will be harmonization at the regulatory level. There are too many other agendas going on here. But, this industry, the regenerative medicine industry, is one of the few that I think, can handle those differences. We do have a few other things we're using this for, and this is just in there. Again, the thing I'll say here is this program, which is really pretty spectacular, was funded by the Defense Department. And Michael also yesterday spoke about how in the U.S., for instance, you have multiple government agencies who theoretically all belong to the same government, all funding different kind of stem cell programs and not talking to each other and nobody guiding it. And with all respect, I think that's a good thing. Right? The last thing we need is one guy or one group of people who have the same academic, background and the same friends and the same agendas telling everybody what they should do for research. I think, in fact, that having DOD, and DOD funded our small molecule regenerative medicine program also, a thing called the Warfighter of the Future, and I'm going to talk about that for a minute. But having DOD fund their own group and having NIH doing their own thing and all the other groups, I think is a good thing. Right? There isn't anybody that knows enough about this field yet for it to be that directed or that guided. And again, I think that's one of the strengths of regenerative medicine is this sort of organized chaos that we all live in. So I do want to talk about the small molecule program, and I do want to leave some time for questions. There are really only two areas in the adult brain where we know that there are neural stem cells, right? And the olfactory bulb area is sort of left over from when we lived by the sense of smell. But the hippocampus is the area that has garnered a lot of research lately. And as I said, we can grow human hippocampal neural stem cells. And so the Army actually gave us $2.5 million to discover a new class of drugs that would be specifically neurogenic in the human hippocampus. Now, while that was happening, there's been a very high correlation, of course, of hippocampal atrophy to a lot of different indications. Psychiatric indications, depression is the one that we're in a second trial now, a 1B to treat. But also you see it now highly correlated to traumatic brain injury and the cognitive as well as the psychiatric downstream consequences of traumatic brain injury. And so I think that the world of regenerative medicine, which has heretofore been pretty focused on cell therapies, whether it's Athersys and their kinds of technology, or ACTC and iPS cells and ES cells or R cells, it's all pretty much been focused on cell therapy. But I think 5 years from now at this meeting, we'll be seeing a lot of small molecule companies that have figured out ways like we have to move these cells. That's probably where this is headed. We think this is the world's first truly neurogenic compound. Again, we're seeing, in addition to the in vitro data, and again that's against human hippocampal neural stem cells, in animals we're seeing up to about a 20% increase in hippocampal volume, which we believe is synaptogenesis, right, the creation of new synapses. So I think this is going to be a big area for the future of regenerative medicine. We are in, as I said, the last dose now of our 1B trial of actual MDD patients. This is 28-day dosing. They stay in the center the whole time. This is a first-in-human trial. We did a human, a healthy volunteer trial, and actually the compound shows remarkably safe and benign side effect profile. I think, we're seeing the same thing. This is a blinded, well-controlled trial. In the 1B, we'll have this data out. We are looking at hippocampal volume increase with MRIs as we go through this. Whether or not we see it with 28-day dosing, the next trial will be 90-day dosing. We'd certainly expect to see it there. But we're looking at everything else that you would typically look at in a well-controlled, well-run depression trial. I should mention Maurizio Fava, who runs the Harvard Clinical Trial Network, is our guru in this area, and designed the trials, and we'll be running the phase 2. So, again, Neuralstem is kind of into regenerative medicine in every space now, right? We're involved in trying to figure out how you can commercialize these as we start to see good human data. We're in trying to figure out how you can get approvals to run trials and then products in, very different cultures across very different continents, and with products that go from small molecule to what you think of as traditional, cell therapy. I think it's rather remarkable that, yesterday there was a guy up here at the Regenerative Medicine conference showing a slide that said chronic tennis elbow. Right? So we're actually, this industry is now going all the way from chronic tennis elbow to having ALS patients, right, who've never, ever shown improvement, going to the beach with his kids and being strong enough to swim and do things again. I think that's a remarkable achievement in a very short time. I think, we heard these guys talking yesterday, the Roche guy and the Athersys partner at Pfizer and a guy from Sanofi. And I have to say, I thought it was extremely disappointing, although I wasn't surprised. The fact is, I think, when you hear a guy who was a Genzyme employee and they get swallowed up by Sanofi talking about the fact that, well, if you have 2 or 3 year data, good data on patients, you really shouldn't go forward because what will happen 10 years from now? We need a lot more work on the mechanism of action and what's going to happen. That's Sanofi swallowing Genzyme whole, right? Somebody who worked for Henry Tamir would never have said that. Okay? I'm serious. I mean, this was a remarkable statement to hear from a guy. So it doesn't take much to see that what's going to happen there is they're going to just defend and try and enhance the billions of dollars of revenue they bought from Genzyme, right, in those markets. But none of the things in that pipeline are ever going to get developed. And the guy from Roche basically said the same thing. He said, well, diligence is diligence. We want to be able to check off the same boxes, whether it's a vaccine, a small molecule drug, a cell therapy. No. Never going to happen, right? That's not the way cell therapy is going to work. Now, there are companies that will do it. The guy from Pfizer said, yeah, we needed to have some comfort level with the mechanism of action. And they did the deal with Gill, right, with Athersys. And so you're going to see companies that will and won't. There's going to be a very, very clear divide. I think that we're fortunate in this industry, things are moving really quickly now. Right? Everybody that presented here showed data, real human data. And think about this. Think about the model that Neuralstem is an example of. We have 16 employees. We're going to be in 7 human clinical trials this year across multiple continents, across multiple indications, all of which are large unmet medical needs. Right? 16 employees. Gill is all over the world, ACTC. These are models where companies are now taking on large unmet medical needs with regenerative medicine products, right? Starting to show really good human data, good intellectual property protection all over the world, and doing it on budgets that, wouldn't have funded the pharma companies' Christmas parties 10 years ago, right? If you put all those budgets together around the world. Serious. Think about that, right? It's a remarkable change, okay? Now, I'm not going to stand here and say that the pharmas are consigned themselves to the dustbins of history, but some of them have, right? And so I think, again, I want to thank the sponsors of this conference for putting this together. The industry is moving remarkably quickly now. And again, to look at chronic tennis elbow up on a slide at a meeting like this is not something anyone would have thought of 3 years ago. And then to see ALS patients actually becoming stable over years, right, and improving, I think we're there, and it's real. So if there's any time, I'm happy to take questions. Thank you.